So I'm going to speak about machine learning to untangle the genome assembly using graph neural networks. And this is a joint work with the Genome Institute of Singapore with Lovro Dweczek, Martin Schmidt, and Mile Sikik, and also Thomas Laurent from LMU, who is in the audience. So here is the general outline of my presentation. So first I'm going to quickly describe the genome assembly task. Then I will talk about the assembly graph. Then how do we decode to get the genome sequence? I'm going to introduce our contribution, the dataset that we used, and then I'm going to show you the technique in detail. Okay, so what is the genome assembly task? So any living organism, human, animal, plant, and so on, so they have a genome. Okay, so what we can do is that we can extract a biological sample, we can give it to a sequencing machine, and then we can solve the genome assembly task that will reconstruct a sequence, sometimes a very long sequence of base pairs of amino acids. Okay, so A, T, and C, G. So for humans, the length of the sequence is 3 billion base pair, but for example, for virus like COVID-19, it's much smaller, so it's 30,000. Okay. So the problem is, of course, very important because the genome contains all the instructions that we need to survive, to develop, and this is something also very important to understand disease and hopefully predict them and cure them. So a quest starting a long time ago to reconstruct the full human genome. So it's starting in the 90s. And the first result was announced actually by Bill Clinton in 2001. So he was at the front page of Nature. And it was announced that the full genome, human genome was reconstructed, but actually it was not fully reconstructed. It was 99 point something reconstructed, but there were still some gaps in the genome. And it took actually more than 30 years to have the first complete genome without gaps, okay, gapless, basically. And it was announced actually last year, so it was also at the front page of Science. And this is, again, the first time that we have a complete gapless human genome. So if we talk a little bit about genome sequencing machines, so unfortunately, there is no machine today that can sequence in one shot the human genome because it's too long and is going to break, basically. So what machines can do is actually to produce collection of subsequences of genomes that are called reads. Okay. So the modern machines, and the goal of these modern machines is to sequence these reads as long as possible by minimizing the errors. Okay. So for example, we have machines like PacBio HiFi reads, so they produce reads subsequences of average lengths of 20,000 base pairs, and the error is 0.5%. Okay, so if you look at 20,000 base pairs, it means that you're going to have 100 errors per read. There are longer reads that you can create, so with the Oxford nanopore reads, and in this situation, the average length will be 75,000 base pairs, but you're going to have much more errors. Okay, so like 5% error, which is going to be 4,000. Of course, what happens is that usually when we want to sequence the genome, we are not going to do with only one read to cover one base pair. We are going to use multiple reads, and this is called the coverage depth. So usually for each base pair, we're going to have 30 reads that are going to cover these base pairs. So this way, we will be able to error correct the reads and be able, to have the best possible clean genome. Okay. But what happens is that so we start from the biological genome here, so the 3 billion base pairs. It will go to the sequencing machine, and the output of the sequencing machine will be a set of unorganized reads. Okay, like this. Okay. So the genome assembly problem is actually a simple problem to state. This is a combinatorial problem, and the goal is basically to reorder the overlapping reads in order to find the longest possible sequences. Okay, so this is how we can reconstruct the genome. And of course, because we have to look at all possible configurations in the brute force scenario, that would be NPR, that would be n factorial complexity. And n is actually very large. So if we take the human genome, n is going to be, so we have the genome length is 3 billion base pairs. The average read length is 20,000 base pairs. We have 30 depth value. We have 2 strands, the 2 complementary sequences, and then we have 2 haploids, one from the mother, one from the father. So n is actually 18 million for humans. So that's really, really large. So going from this set of unorganized reads to a complete genome is very hard combinatorial problem. So we need to reduce the complexity of the problem. And the first step to reduce the complexity of the problem is to construct a graph. So I already told you that what is important is to look at the overlapping reads. Okay? So when I talk about overlapping, then I can think about an edge between 2 overlapping reads, right? And this, an edge is going to, the collection of edges is going to be a graph. Okay, so I have this genome, so this is the one that I'm looking for. The machine is going to produce these unordered reads. And then what I can do is that I can look at, the overlapping reads. So I can look at this read, and this read is going to overlap with this read here. Since they overlap, I'm going to make a connection, okay, 1 and 4. This read 1 and 4 are going to be connected, so I'm going to put an edge, okay. 3 and 4, they overlap, so I can also place an edge here, and so on and so on. So this way I can construct what we call an assembly graph, okay. And in genomics, this is called the overlap phase, okay. So this way we can reduce the original complexity to the complexity of the number of edges in the graph.
Xavier, you have mistakes in the reads as well, right?
Sorry?
You have mistakes in the reads as well.
Yeah, you have errors also in the reads, absolutely. So, and I'm going to talk about this, of course, yeah.
What does the direction mean?
The direction means that when you look at the overlap, you can have an overlap between this read and this read here, but for the same reads, if you put it there, you may not have any overlap. The direction just means that you are looking at this read in this direction and this read in this direction. But it's not true if you reverse the 2 reads. You may not have any overlap. Okay. So the challenges are basically, so you may have interspersed regions, which means that if you look at, this is the genome, okay, and this is the complementary genome here. So they are the two strands. If you look at one read on the genome, it may happen, so, and you look at this read as well, so these 2 reads are going to overlap, and they are consecutive on the genome, okay, so everything is fine. But you can imagine having another read, which is exactly the same as this one, but far away on the genome, okay, so you will have actually a wrong connection between this read and this read here, okay. And the same can happen also on the negative strand. So you may have a read which is exactly the same as this one, and suddenly you are going to create a wrong connection between this read and this read here. Okay. So what will happen is basically you will create, for example, a shortcut here, or you may create a connection between 2 different strands on the genome. So this is going to be bad. But actually, the most important issue is the segment duplication. So what happened is that in the genome, there are regions with a lot of repetitions. This is probably related to the robustness of the genome. So, for example, if I'm looking at this genome, if the read is this length, okay, what happened is that I'm going to produce a lot of repetitive reads, which are exactly the same, which means that I will create actually a bundle of connection between the reads. And in this situation, I will not be able to disentangle, the correct path inside. So this is actually a major issue. And today, there is no solution to that. So what people do, basically, if they encounter this situation, they will just, cut the genome, and they will have fragments of genomes that we call contigs. And that's the best we can do. So the solution can be either from the machine. So imagine that we are able to create a machine with a read which is larger, than this region, then we will solve the problem. Or the other way is to have an algorithm that is able to disentangle, the correct sequence in this bundle here. So
the problem, I assume, is that you don't know how long is this sequence in order to cut. Right?
No, we don't know. Yeah, and I'm going to talk about this. It's basically the combinatorial part. So you want to construct the longest possible sequence. Yeah. Now, let's talk about the construction of the graph. So if we want to have an exact construction, the complexity will be basically, n squared, d squared. N is the number of reads, and d is the dimensionality of the reads. Okay? Because for each read, you need to compare with all other reads. Okay? So you have n. And then you need to look at, the overlap. So you need basically to look at, the shifting. So it's going to be d squared. So the complexity is really huge, actually. N for human genome is going to be 18 million, and D, if you take HiFi reads, is going to be 18,000 dimensions. So something interesting is actually, if you do the convolution between two reads, it's very, very fast today with GPU. The issue is basically to transfer all the reads from the hard drive to the GPU memory. So this is where it would take too much time. But we can imagine that, maybe in a decade, that something is going to be very, very fast. But today, we cannot do an exact construction of the assembly graph. So we need approximation. And in the literature, there are as many approximations as, genome assembler. So each team is going to develop its own, approximation of the graph. And also, depending on the type of reads, so depending on the machine, if this is HiFi reads, if this is nanopore, you may also have different ways to construct the graph. So that's a lot of variations. So if I put everything together, so if I'm looking at the sequencing errors, interspersed duplicated regions, and then approximation of graph, it makes the topology of the assembly graph very, very challenging, actually. So you may have disconnected components coming from the bundles, cycles, dead ends, bubbles, transitive edges, and tangles. So that's a complicated graph topology to solve. So once you have the graph, then what you want to do is precisely to extract the longest possible path on this graph. So this is a path routing problem. And you want to do that by visiting each node at most once. You're going to have a lot of actually cycles. So you want to avoid that, okay? Once you have so for example, for this illustrative example, this is, for example, in red, one of the solutions was the possible longest path on the genome graph, on the assembly graph. Then what we're going to do is that each node represents a read, and then between these two reads, basically you are going to concatenate all the reads, okay? And this way you will be able to reconstruct the full sequence, okay? This decoding step is called a layout phase in genomics, okay? But as I told you before, is that the problem is usually these graphs, they are very noisy and they are disconnected. So in practice, the best we can do actually is not to reconstruct a full sequence, is to reconstruct, some part of the sequence. These fragments are called contigs. And the goal, basically, when we compare genome assemblers, we are going to compare, the algorithm in terms of the length of the contigs and the quality of reconstruction of the contigs. That's how we compare algorithm. Okay. So here, this is one of the state-of-the-art technique for the genome assembler. This is called Raven. And basically, this is based on a set of engineered rules. And the idea is to start from the noisy and large assembly graph and then simplify it. Simplify it until we get one sequence. Okay? So usually we can get one sequence, we're going to have multiple sequences. So Raven heuristics are this list. So the first one is we are going to remove transitive edges. Okay? So if we go from this read here to this read here, usually we are going to remove all these transitive edges and we are just going to keep these edges here. Why we want to keep these ones is because we're going to have a maximum overlap, and this will help us, to reduce the error of reconstruction. The more overlap, the better for the reconstruction. The second rule that we are going to use is we are going to remove all dead ends. Okay, so again, the graph is noisy, so we may have this situation, so we are going to remove it. We can also remove bubbles. So usually it comes from the positive strand and the negative strand. So we are just keeping the path, which is the longest one between these two. Fine, and then we can collapse, long sequences into like a supernode, a unitig, like this one. The next step is to do a dimensionality reduction in order to remove long connections between the reads. And finally, we are just cutting the sequence when we identify tangles. Okay, so the output will be a set of contigs, a set of sequences. That's what we can do. Okay. So, so in 2022, so last year, again, that was the first time that we get a gapless complete human genome reconstruction. And the success is basically, achieved because of the machine. So we have machines that are able to produce longer reads with less errors. Then we have a combination of multiple genome assemblers. And finally, because we don't have any genome assembler to understand, the tangle, we have human experts looking at, this tangle and trying to find the correct solution. So this is how it is done. So we have experts looking at manually and, to resolve these tangles. The limitation is obvious. So this is a time and resource consuming, and this is not generalizable. So what we would like to do is to use machine learning to solve this problem, either by reducing the human heuristics or doing everything end to end. That's the future. And the goal is basically, suddenly we will not depend, yes?
So before you go into your contribution, can I ask if any of these techniques are actually using the genome of multiple people? Because there is a lot of similarities between them, obviously.
Yes.
And so you could actually find out where are the different pieces located, right?
Yeah. There are algorithms that do that, but it's even more difficult because then you need to identify, different genomes for different person. So there is also many techniques for example, microbes. So they want to do that to identify different microbes. The problem is even harder to do that. But yes, there are some techniques that try to do that. The problem is not to identify reads. The problem is to reconstruct the sequences and separate, individual sequences to each other. That's the hard part. Yeah. Yes.
So I'm not sure why you're using just a directed simple graph, like the assembly graph. If you really care about sequences, why not use like the growing graphs or a higher order network?
Oh, you can use any structure you like.
No, but
so
that
here
will make your machine learning harder.
Sorry?
Right? That will make the application of your machine learning harder, right? So I want to know whether the choice of a simple directed graph was because it makes the machine learning easier down the road, as opposed to using something like a higher order network, like a De Bruijn graph, or other types of network representations that can hold memory.
So for the De Bruijn graph, this is something that we would like to try next because it has a lot of mathematical properties, symmetries that we could use in the GNN, the architecture of the GNN. Yeah. So some in the team we developed. So they are in contact with San Diego. So they are doing, they are expert in this type of graph. So this is something that we would like to investigate. So far, we are doing, I would say, the simplest, approach. We are using the graph construction developed by the team I'm collaborating with. But you're right. I mean, the more structure, the better. So here we don't have any special structure. It's just if we have two overlapping reads, we have an edge. But we could use probably, more structure properties to improve the decoding. I agree. All right. So one thing that we're going to do. So again, there are two parts in this problem. The first part is to extract the graph, the assembly graph. And the second part is to extract the path in the assembly graph, okay? So obviously what we would like to do is to learn everything end-to-end, right? We would like, given the set of reads, we would like to learn the best possible graph, and then we would like to decode it, okay? Find an agent that is able to decode the longest possible sequence. This is very hard. So what we try to do first is to use an existing graph constructor, which is given by Raven. And then from this graph, basically, we want to learn an agent that is able to decode the longest possible sequence. Okay, so we make an assumption that the graph is given to us, and then we try to, from the graph, learn an agent that is going to decode the longest possible sequence. But of course, we would like to do everything end-to-end. Okay, so this is the machine learning framework that we introduce, and I'm going into the detail of this picture. So first, the data set. So the good news and the bad news. So the good news is that so we have a genome, a human genome data set, okay, and this is completely clean, okay. So this is the CHM13 human genome sequence. So this is the one that was in the paper, in the Nature paper. This is one haploid coming from the mother, 23 chromosomes, 2 strands, and it has 3.3 billion base pairs and 5.6 million HiFi reads. Okay. So we improve a little bit, by reducing the error and also by mapping basically the reads to the original sequence, because we want to know the position of the reads if we want to be able to know the correct solution of our algorithm. But something which is a limitation is that today there exists only one clean human genome, only one. So this is an issue because in machine learning you would like to have a training set and then a test set, right? So we just have one genome data. So what we're going to do is that to validate our technique, we are just going to use not the genome, but we are going to use the individual chromosomes. And we will have 2 sets, one for the training and one for the testing, and also another one for the validation. But this is one of the limitations of what we do, and we are trying, to collect more human genome. But this is hard because they are not clean. So this is something to figure out. For the data augmentation, so we need to do some data augmentation. And for this, we are going to simulate reads from the chromosome. So how we do that? Basically, there exists some read simulator. And the thing for this simulator that we use here is that it doesn't have much constraint. The only thing it wants to keep the same distribution of lengths between the real reads and the artificial ones. Okay, so this is the only constraint of this simulator. And this way, so if we have one chromosome, we can simulate many other chromosomes, actually many other reads of the chromosome this way. Okay. And there is something which is very important, is that because we simulate the reads, we know exactly the position of the reads. So for each read in the chromosome, we know their position, we know their ordering. And this will be very important because this will be our supervised signal to train the network to reconstruct the sequence. So this is the important part for the machine learning algorithm. And again, I want to emphasize that we do that for individual chromosomes. We don't have like one genome for training and one genome for testing. This is not something that we have today, hopefully in the future. So, yeah. So, for the graph construction, again, we are using the overlap phase of Raven. So this is the team I've been collaborating with. And basically, this is composed of two parts. The first part is going to do a dimensionality reduction. So you start with a read which has 20,000 dimensions, and you need to reduce it. Otherwise, you will not be able to compute, all pairwise comparisons. So the first step is basically dimensional reduction. You go from 20,000 to 512 dimensions. And basically, the idea is to extract repetitive words inside these reads. And then the second step is, of course, to do the pairwise matching comparison between the reads. And for this, we use a standard, algorithm to solve this. So for a chromosome of 50,000 reads, it's going to take 20 minutes, basically, to reconstruct the graph. And this is our pipeline for the data set. So we have the THN13, one chromosome. We extract simulated reads. Then we use Raven to construct the training set of graph. Okay, and here and we use also Raven to construct the test set of chromosome graphs. Okay. Okay, so to decode, we need to have an algorithm that is able to identify the correct age on the assembly graph. Okay, so if this age belongs or not to the solution, that's what we need. So we need a labeling mechanism to identify if an age is part or not of the solution. So something that we have, because we have the clean genome, and we already map all the reads to the genome, so we know exactly the positions. Okay, so we can use the position. We can extract the position using any search algorithms. Here we use DFS, but any search algorithm will do the job. And basically, we can label the correct and the bad edges. So if we have this assembly graph, all the nodes which are red, which will have a label of value 1. Okay, basically it's correct. Okay, if you follow this path, then you will be able to reconstruct the genome. Okay, if the wrong edges will have a blue value, right? So here, this is for example, a dead end, or maybe this is a shortcut. So these are going to be labeled with the value 0. So we don't want the network to use this, this edge here. All right. So we need a mechanism to predict edges now. So we have the assembly graph. So we need something to decode. We need an algorithm to say, okay, what is the next edge in the solution? To do that, so first of all, we need to identify which class of networks we need to use. So the problem is fundamentally a graph, right? So I already talked about if we have an overlap between two reads, we're going to have a graph, right, an edge. And also the decoding is done on a graph. So I'm going to use something which is not a CNN, because CNN only work for grids, RNN work for sequences, and transformer works for fully connected graph. But I don't have a fully connected graph. I have a directed graph in this situation. So I'm going to use a graph neural network here. Okay. So now I'm going to talk about the design of the graph neural network. Okay. So what network properties I need? So I will need a standard, property. I need my network to be invariant with respect to the permutation of the indices. Okay. I also want my network to be independent of the number of neighbors I'm going to have, and also the size of the graph. I can have a chromosome with different sizes. I want to be independent of that. Because my graph is directed, I would like to have some anisotropic diffusion process. Okay, so And the local reception field is also directed. Okay, deep architecture, the deeper the better. And I'm going back to that. So basically, you know that a GNN is basically composed of these two update equations. The first one is going to update the node representation in the graph. So this is layer L. This is the next layer, L plus 1. To update the representation of the node, I'm going to use the neighbors and also the current representation. And also for the edge, I'm going to use the previous edge representation and also the two nodes that compose the edge. And then you have you have a large literature to decide what is the best function for the node update and the edge update, basically. So here we use something that is based on actually peronomatic. So this is a technique that was designed in image processing to denoise images. So in image, you want to preserve edges, okay? And you want to smooth out the noise in the homogeneous region. So you need to have an anisotropic diffusion process. So this is something that we use here. We discretize peronomatic on the graph. And we had also directed edge information here and here. So the graph is directed. This gives us a little more information, a little more inductive bias. So for the edge, coming to this node, and the edge outgoing, basically. So we have more parameters, so we have a better GNN to predict the edges. Okay. So of course, the model is permutation invariant, and also we can distribute this GNN model using standard libraries developed for GNN, like DGL or PyTorch geometric, the one we like. Okay. The local reception field is directed. Again, for this node, this read here, we have predecessors, and then we have successors, and we can differentiate both when we make the prediction. And then we have additional tricks for the GNN to learn well, which is batch normalization or usual connection. And then I'm going to talk about the node feature in the next slide. So first, the edge feature. So the edge feature is very obvious. Basically, we want to look at the length of the overlap between two reads, okay, and also the quality. So sometimes we're going to have errors. So we want to use this error measurement into the input feature of the edge. For the node feature in this work, we do not use any information coming from the reads. So in this situation, they are what we call anonymous. And we know that GNN perform poorly when we cannot identify the node, okay? So there are different ways to fix the problem. One way is to add positional encoding, okay? So one positional encoding that we introduced before was Laplacian eigenvector, but this is for undirected graph. Now we are working with directed graph. So the one that is very natural to use is PageRank. Okay, so we use a few steps of PageRank diffusion as positional encoding. We also add the in degree and out degree, in the node feature. Okay, so for the input feature, so we start with the raw node feature and the raw edge feature. We do two small MLP to project into a higher dimensional space. And then we do the convolutional layers. We do L number of convolutional layers. So this is for extracting the features. And then we are going to make the prediction. So the prediction is basically, we have this assembly graph, and we would like to know if there is a connection between I and K, which belong to the solution. Okay, so we have the node I, the node K. So the way we make the prediction is simple. We just do an MLP, and where we concatenate the node representation of I, the node representation of K, and also the edge representation. We just put everything together, and we have sigmoid to have a probability between 0 and 1. So very simple. Okay, and then we train in a standard way. So we have 16 layers, 256 dimensions, 256 hidden features. So it gives us a network of 6.5 million parameters, and we use cross entropy. to train this network, because we know the ground truth, basically given by the positional information of the reads. All right. So for the size of the chromosome graphs, so they vary between 32,000 to 200,000 nodes. So they are usually too large to be contained on the GPU memory. So what we do is basically we use graph partitioning to cluster the big chromosome graph into smaller graph. And for this, we use METIS, which is a clustering algorithm which has a very good trade-off between speed and accuracy. Okay. So for the graph decoding, what we want to do is to extract a sequence of nodes on the assembly graph, which is going to maximize the probability, so the length of the extracted sequence. We can use the chain rule of probability. The probability of having the next node will depend on what we have before, basically. So we use greedy search here because it's very fast. We just use the previous node. The complexity is linear. But something that we do, even if it is fast, it is not enough because the graph, again, is very noisy. So what we decided to do is to actually use k initial nodes that we sample by using Bernoulli. So we have the probability of each edge. So given this probability, we can, extract k edges by Bernoulli, okay, and then we use that as an initial edge for the graph reconstruction. So we go forward and then we go backward, and this way we are able to reconstruct one genome sequence. And we do that for k path, and we select the longest one. Okay, so again, the graph is very noisy, so this is something very important to do. So we repeat that until we are not able to extract a path more than a certain length. Okay, so we stop, and that's it. Okay, so for the experimental results in setting, so again, we are working with individual chromosomes. We train using one real chromosome and only one. So chromosome 19, it's a chromosome which is not too complex and not too easy at the same time. But we try with different chromosomes and also a set of 3 chromosomes. More or less, the results are the same. Okay, so usually one chromosome is enough for training. So we use one chromosome for training and the rest for testing. For this chromosome, we are going to generate 15 synthetic graphs and 3 validation graphs, basically. So we train, and then we stop when we get the lowest validation loss on this tree graph. And the training takes like an hour, basically. Okay, for the inference, so once the network has been trained, inference is basically doing the forward pass and then the greedy decoding. Yeah. So to evaluate the performance, so in this domain, people use different measures like the number of contigs. So remember that we cannot extract one sequence. We are going to have fraction of the genome. So we want to have, the minimum number of contigs. Then the length of the longest contig, okay? The higher the better. The genome fraction, so how much percentage of the genome you are able to reconstruct. Then you have NG50. So basically, if you put all your contigs together and you're looking at 50%, you take the contig which is in the 50%. Okay, so the higher the better. If you have the whole genome, and this is the one that we have, we take 50% of the true genome. Okay, and this is the length of this contig here. So again, the higher the better. And then finally the base error. So how many errors we are doing in insertion, deletions in the reconstruction of the genome. Okay. So this is the result. So here you have the machine learning part, and here you have the heuristic algorithm. So when it is in bold, it means it is better. So here you have the chromosomes, 1, 2 to chromosome X. And you can see that the machine learning is able to globally to do better than the heuristic algorithm. And this is also true for the mismatch and in there between the machine learning and the heuristic one. Okay. So, so as a conclusion, so this is just the first step, right? So the graph, we didn't learn the graph is given to us by experts. But for the decoding part, when we extract the sequence, then the machine learning technique is able to outperform the engineering one. Okay, so this is just a first proof of concept. Something that is interesting is that we release the data set. So in the community, in the GNN community, usually we use small data sets, and they are not really real world. So here we try to also release a graph of chromosomes, which are not small, and they are from real world. So that may be also a new data set to compare the GNN architectures. Yeah, okay. So the next steps is, the first thing we want to do, and we are working on that now, is to look at different graph constructor. Okay, so as I said, there are different ways to construct the graph. So we would like to try for different type of graph. If the GNN is able to do better than the heuristic one. We also want to evaluate on the whole genome, not only on individual chromosomes. And this is also something that we are working on. So we have access to additional human genome, but they are not as clean as CHN13. So we need to find a way to make, a fair comparisons between the techniques. We need 2 haploids. We are only 1. We want to do something different from humans. So in Singapore, they have a national program that is going to sequence 1 million of genome from the population. And the idea is to use that for healthcare and predict, future disease and, prevent better. Yeah, and of course, the ultimate goal would be to learn everything end to end. We want to learn the graph, but also we want to decode at the same time. Thank you very much.